All right, we're live, so you can talk.
Great, thank you. Okay, so welcome back to Cosyne. This is session 3, and we have 3 great speakers that will be sharing their work with us today. Please post your questions to Slido and feel free to vote for other questions that you would like to have answered. So our first speaker today is Cooper Grossman. Last week, Cooper successfully defended his thesis in Jeremiah Cohen's lab at Johns Hopkins, so congratulations. And today he'll tell us about his work studying serotonin neurons during value-based decision-making.
Great, thanks Christine for that introduction. Let me just open up my presentation here. Okay, can you see that and hear me okay?
It looks good.
Okay, great. All right, so I'm Cooper Grossman. I am a member of Jeremiah Cohen's lab at Johns Hopkins, and today I'm going to talk a little bit about the work that we've done investigating the role of serotonin neurons in the processes of learning and decision-making. So the brain faces this difficult cognitive task of learning about the consequences of its action, and it's difficult for several reasons, 2 of which I'll describe here because they're relevant. So the first is that the outcomes of actions are noisy, they're probabilistic. So the question is then, how much should we learn from any individual experience with a single action? One strategy is to learn an estimate of variability about that action, which we're going to call expected uncertainty, and use that expected uncertainty to downregulate the amount that we learn from each experience. And we want to do this in stable environments because we don't want our behavior to be driven suboptimally by stochastic fluctuations in outcomes. But we also need to be able to detect change in the environment, which results in a change in that action-outcome contingency. So here we can generate an estimate of unexpected uncertainty in order to enhance learning rates. And we want to enhance learning rates because it's now only the most recent outcomes that have useful information about this relationship. And so work from human studies has shown that behavior can be characterized using uncertainty-driven learning, which I'll call meta-learning, in this way. And so we wondered if mice engaged in any sort of meta-learning and leveraging the tools available in mice to see if any reflection of that process can be found in the neural activity. And so we modified this task, a foraging task in mice that was originally adapted by Bilal Bari in our lab from a monkey task used previously. So in this foraging task, the animal samples, head-fixed thirsty mouse samples 2 potential sources of water that are probabilistic and dynamic. And now those states are not signaled to the mouse, so the mouse has to rely on its action and outcome history in order to behave adaptively. And so we spread decisions out in time using this trial-wise construction. On each trial, the animal receives an odor go cue telling the animal it can make a decision, but otherwise gives it no information. The animal can then lick either the left or the right spout, and as a consequence of that decision lick, the outcome is delivered probabilistically from the chosen spout. Now, the probabilities assigned to either spout are drawn from a set of 3 discrete probabilities. So here, 0.9, 0.5, and 0.1. And those probabilities are assigned independently to each spout, as we can see from this example session. And so what this does is create a rich set of reward dynamics that we can then use to test ideas about how those reward dynamics shape learning, if they do at all. And so overall, animals do quite well in behaving in this task. So this is just an example session from a single mouse. On top of the y-axis, the lines indicate a choice to the right, and lines below a choice to the left. So the long black ticks are rewarded, and the short gray ticks are not rewarded. The black line here represents a moving estimate of choice probability that we can get simply by smoothing over the raw choices. And when we do the same for outcomes shown in blue, we can see that the choice behavior dynamically tracks the outcomes over the course of the session, even as the probabilities of reward assigned to each spout are changing with relatively high frequency. And so this shows, among other analyses that we did, that animals are learning from outcomes in order to drive choice behavior. But we also wanted to see if the rate at which they were learning was indeed variable. And so we looked at these rare instances in the task in which the reward probabilities assigned to each spout change simultaneously or within a few trials of each other. So 2 of these conditions are plotted here. The black line shows high and low assignments being swapped to low and high. And the gray line shows medium and low assignments being changed to low and high. So prior to the transition, we can see that the animals choose the high side more often than they choose the medium side, as you'd expect. But the more interesting feature is that after the transition, even though the animals are both adapting to the new high side, they adapt more quickly coming from this high background as opposed to the medium. And so to make sure this was an effect of the difference in outcome history and not the apparent difference in choice history, we limited this analysis to instances in which the animals exclusively chose either the high or medium spout for the 10 trials prior to the transition. And you can see that even in this case, coming from the high background, the animals adapt their choices much more quickly. So we also wanted to look for evidence that maybe this potential variability in learning rates was driven by uncertainty. And we wanted to do this first in a modelless way. So we looked at, we did one more analysis looking at these medium transitions from the right plot here, but now we're going to sort it by reward history. So the idea is that when the reward rate is exactly equal to 0.5, we're going to maximize the variability and thus the expected uncertainty of the outcomes. But as that reward rate either increases or decreases, then we should be decreasing expected uncertainty in both of those cases. And so the cyan line shows that low reward history condition. The magenta line shows the high reward history condition. And in both of these cases, the animal adapts their behavior much more rapidly than when that reward rate is exactly equal to 0.5, at least consistent with maybe the animals using uncertainty to guide learning, the way described earlier. So to formalize this in terms of a generative model, we turn to the classic family of reinforcement learning algorithms called Q-learning. So in these models, the agent learns the expected value of an action denoted by Q, L for left, R for right. It can then compare the expected values in order to make a decision, and then update the expected value of the chosen action by comparing that value to what actually happened, the outcome. So this difference, of course, is the reward prediction error, and that's used to scale the old expectation at a rate that's controlled by alpha, the learning rate parameter. And so typically this parameter is treated as a fixed parameter, which means that the rate at which animals learn from outcomes is going to be static across the entirety of the session. And so this is what we're going to call static RL and use it for a baseline to compare our meta-learning modification to. So incorporating our ideas about uncertainty into this model then, we're first going to change this alpha parameter to a variable, and we're going to downregulate that learning rate as a function of expected uncertainty, epsilon. And so we're going to define expected uncertainty as the history, recent history of reward prediction error magnitudes. So basically, how far off on average were our expectations from what actually happened? But we also want to be able to detect potential changes in the reward contingencies. So we're also going to allow unexpected uncertainty, upsilon, to upregulate that learning rate. And the way that we derive unexpected uncertainty is going to be the difference between the absolute value of the prediction error and the expected value of the absolute value prediction error, or expected uncertainty here. And so unexpected uncertainty can kind of be viewed as a prediction error that tracks deviations from the typical variability in order to enhance learning. And much in the same way that we use the prediction error to update the expected value, we're going to use unexpected uncertainty to update expected uncertainty. And so we fit both the meta-learning model and the static learning model to behavior. We get the parameters from those fits and plug them back into their respective models, and then simulate behavior with those agents in order to see if the simulations can capture what the animals are actually doing. So those simulations are shown in this green rectangle here, and we can see that the static learning rate fails to recapitulate this apparent difference in learning rates across these different transitions. The meta-learning model, however, is able to capture this apparent difference in learning rates. And so one way we can quantify this is just by looking at the relative slope of these choice average lines here, and plot the time constants, which will give us sort of an estimate of their steepness. And so that's what I'm showing in these bottom panels here. And in the static learning model, you can see that the relationship between time constants does not match what we see in the actual data given by this black dot. And here, each of these green circles represents simulations from an individual animal running these experiments. The meta-learning model, however, is able to capture this relationship, indicating that the high-low to low-high transition is steeper than the other. And so an obvious question is then, if we have some behavioral evidence of this, is there anywhere in the brain that could be conducting meta-learning? And so theories have long proposed that neuromodulators sit in a prime position to mediate meta-learning because of their massive innervation of regions associated with learning and decision-making, their activity on behaviorally relevant timescales, and their response profiles to valued outcomes. So we focused on serotonin in particular, because manipulations of those neurons have showed effects on learning rate without actually abolishing learning, about a change in contingency between action and outcome. And so we recorded from the dorsal raphe of mice behaving in this task, and we selectively transfected serotonin neurons with the light-gated ion channel chanorodopsin under the control of the serotonin transporter gene. That way, at the end of each session, we could identify any recorded neurons as serotonergic by their response to brief pulses of laser stimulation with short latency, high reliability, and the matching of the evoked waveform to the spontaneous waveform. And we first looked at the activity of these neurons over long time scales, so during the intertrial interval, or ITI. And what we found is that slightly more than half of neurons, their activity correlated with this model-generated expected uncertainty term, which again is this moving history of prediction error magnitude. So in these two example neurons on the left, we can see their ITI firing rates plotted in black and gray show decent correspondence with the behavior model-generated expected uncertainty shown in blue. And as you can see by the sign here, these neurons are both negatively correlated, but across the population, as we can see in the t-statistic distribution, we see both negative and positive correlations. And so these are the averages of those two different populations, and we can collapse across the two by sign flipping the positively correlated neurons, and that's where we get this relatively linear relationship across the population between firing rates and expected uncertainty. So this is looking at the activity of neurons on long time scales, but one question is, how is that representation updated within the trial period? And the model itself proposes a mechanism by which unexpected uncertainty updates expected uncertainty. So we looked at the phasic activity within the trial to see if that was indeed the case. And so here I'm showing activity aligned to the onset of the cue. So as we can see from the reaction time CDF, this is prior to the receipt of the outcome until later on out here. And we're looking at the firing rates graded by the value of expected uncertainty relevant for that trial. And so you can see by the different shading of these colors that the representation is pretty clear, but as we get closer to the outcome, on average, that representation starts to get a little muddy. But when we look at the updated value of expected uncertainty aligned to the outcome, we can see that prior to the outcome, this new value is not really well represented, but afterwards, we see this emergence of the very clear separation of expected uncertainty values by the firing rates as a function of receiving that outcomes. And so we also looked at the unexpected uncertainty, and during this sort of expectation-like phase, we didn't really see very nice representation of unexpected uncertainty from the previous trial. But again, when we look at the unexpected uncertainty that is a function of the outcome on this trial, then we see this sort of late emergence of representation across these neurons. And so this suggests, at least on average across the population, that maybe these neurons are updating their expected uncertainty estimates sort of in the manner proposed by the meta-learning framework. So we also wanted to test the idea of what would happen to choice behavior if we were to remove this uncertainty component, uncertainty-driven learning component from the model. And so we fit our model to behavior and simulated a whole bunch of behavior again. And then what we did was to lesion the meta-learning component of it, but leaving all the other parameters intact, and then simulate behavior again. And so that's what I'm showing in these two panels. On the left is the intact behavior. And here it's clear we have the crossing of the choice average curves indicating variable learning rates. But when we lesion the uncertainty component, it basically just becomes a static learning model, and we no longer see that interesting transition phenotype. So to test the idea that serotonin neurons are the ones mediating the serotonin, mediating this uncertainty-driven meta-learning, we selectively transfected serotonin neurons in the dorsal raphe with the inhibitory DREADS receptor HM4-DI. And then we could administer systemically either a control vehicle injection or the DREADS ligand agonist 21 to effectively inhibit serotonin neurons during the entirety of the session. And so this is the actual behavior from 5 mice run through these experiments. And on the left, we can see that the learning rates seem to be variable in the same way as the simulated behavior. but that seems to be largely abolished when serotonin neurons are inhibited. And so again, we can quantify this by looking at the steepness of these choice average curves using exponential fits. And so here are the results from the 5 mice using the simulated data and the simulated lesion. And pretty much in all 5 cases, this relationship between the steepness of the 2 curves changes such that they no longer seem to be much different from one another as they all kind of approach the line of unity or cross it. And indeed, that is what we see when we fit those exponential curves to the actual behavior, which is shown in this right panel here. So it seems like the behavior, the electrophysiology, and these chemogenetic experiments at least are consistent with the possibility of serotonin neurons tracking uncertainty in order to modulate how the brain learns adaptively about the consequences of its actions. And so briefly, I'd just really like to thank the wonderful humans of the Cohen lab, especially Jeremiah and Bilal, who have made substantial contributions to this project. Thanks to the organizers for allowing me to talk. And thank you for your attention. Feel free to reach out.
Great. Thank you so much for that talk, Cooper. That was wonderful. There are quite a few questions. So I'll start with one from Annegret Falkner, and I apologize if I mispronounced that. Is the dependence of learning rate on the uncertainty a model assumption, or does it result from the normative model?
Yeah, so this is a model assumption in the framework that we're using reinforcement learning algorithms, but it is based on normative models of learning in similar circumstances that use things like Bayesian inference to make decisions. So this is sort of trying to be an approximation of a normative approach.
Great. Rabiba Wang asks, if serotonin neurons correlate with expected uncertainty, what is the neural substrate of unexpected uncertainty?
Yeah, so we saw a little bit of a correlation of unexpected uncertainty in the phasic activity of serotonin neurons. So about the time of the outcome when you would expect to see unexpected uncertainty. But yeah, there have been, of course, other proposals about other neuromodulators that could potentially signal unexpected uncertainty, like acetylcholine or norepinephrine. So I don't know of any results off the top of my head, but I do know somebody in our lab is working on the norepinephrine approach. Yeah, and I should also clarify too, the type of uncertainty that I'm talking about here can be thought of as applying to the entirety of the behavioral policy. So the uncertainty is not about a specific action, but generalizes across the actions. So that might be what distinguishes the activity of serotonin neurons from, say, norepinephrine or acetylcholine, which might care about the uncertainty specific to an action or stimulus.
Great. And I think the last question, for the sake of time, is from Anne Churchland. So she asks, did neural responses also correlate with action values? Action values should change with uncertainty, for example, via Thompson sampling.
Yeah, no, that's a really interesting question too. Yeah, so there was a little bit of like relative value encoding in the dorsal raphe, not as much when we just looked only at the serotonin neurons. But it is an interesting idea. And we're looking currently at the effects of serotonin in the prefrontal cortex, where previous work in our lab has showed representation of relative values and total values in a very similar task. So it would test this hypothesis that I think was referred to in the question, that serotonin can actually directly update these decision variables in the way proposed by the model.
Great, thank you so much. So there are quite a few questions. I don't know if there will be a way to answer them offline later, but I think for the sake of time, we should move on. But let's thank Cooper again for a very interesting talk. So our final speaker who will be joining us is Avinash Baidya. Avinash is in Mark Goldman's lab at UC Davis. Maybe I should wait for him to join. There's Avinash, great. Hi. Great, so Avinash is in Mark Goldman's lab at UC Davis, and today he'll be telling us about work that he's done in collaboration with Ilana Witten's lab, studying choice-selective sequences as a possible substrate for credit assignment. So take it away, Avinash.
Okay, so you can hear me and you can see my slides?
Yes, looks great.
Okay, cool. Hi, everyone. So this is Avinash. I'm a fourth year PhD student at UC Davis, and I work in my Goldman's lab. Today, I will talk about how neural circuits in the brain could solve temporal credit assignment problem. So let us consider this example where a mouse can either choose left or right, and depending on what choice it makes, it may or may not get rewarded after some time delay. The question we are interested in is how does the neural circuitry in the brain learn to associate an action to a delayed outcome? Well, to solve this problem, the animal needs to learn the value or expected future rewards associated with each action. And well, value is learned using reward prediction errors or the error in expected future rewards. Let us look at an example. Say you make an action at the start of a trial, and then before learning, you have no expectation of reward. So the value or expected future rewards is just 0 at all time. Now, when you get a reward, you will get an error signal at that time as you were not expecting any reward, which will then update the value function to indicate that the action leads to future rewards. Now, this is a standard problem in reinforcement learning theory. The question is, how does the brain keep a memory of what action was made through the delay and maintain a persistent value signal responding to that action? Here is what we know about the neural circuitry. Previous research suggests that striatum carries a value signal and that the dopamine neurons carry prediction error signals. The key gap in our knowledge, however, is what the cortical inputs to striatum encode in a choice task. The reason this is an important gap is that
I'm sorry to interrupt. Occasionally your audio is kind of going out. Can you try turning off your video and seeing if that improves, just so that we can hear you a little better? Thank you.
Yeah, sure. Okay. So the reason this is an important gap is that we think this input should provide the information that dopamine could modulate to produce value signals in striatum. So our first goal is to record and determine what these cortical inputs to striatum encode. We will then build our circuit model of learning based on these recorded cortical inputs. And experimentally test predictions of our model by perturbing these cortical inputs. Let us start with talking about the behavioral task, which is a reversal learning task. The task for the mouse is to choose the lever corresponding to the higher reward probability. So the trial starts with the mouse poking its nose into the central port. Then after a time delay, it's presented with levers on either side. The mouse then chooses to press left or the right lever. So now the trials are organized into blocks in which either the left or the right lever press has the higher probability of getting rewarded. In a left block, the left lever will correspond to the higher probability of reward, and in a right block, the right lever will correspond to the higher probability of reward. The challenge in the task is that the blocks switch randomly after a variable number of trials. What we found was that the mice are able to switch choice preference very quickly when a block switches, as shown here. So on x-axis, we show the trials before and after a block switch. 0 is the trial when block switches from left to right. And y-axis represents the probability of choosing left, which is the black curve, or choosing right, which is the gray curve. As you can see, on the left block, mice choose left much more than right, and then switch over the course of a few trials to choosing right more on a right block. Now, to inform a neural circuit model of how the mice are able to do this task, we recorded the prelimbic cortical inputs to nucleus accumbens, which is a part of the ventral striatum. So from here on, we will refer to prelimbic cortex as PL or just cortical inputs, and nucleus accumbens as NAC or NEC. Here is what we found. So we found that the cortical inputs show a particular pattern of neural activity, which we call the choice-selective sequences. X-axis here represents time from lever press. 0 is the time of the lever press. And y-axis are the recorded neurons. So one horizontal slice represents a recorded neuron's activity over time during a trial. Now the figure on left shows the average activity over time, average activity over all left choice trials, and the figure on right shows the average activity over all right choice trials. You can see that on both left and right choice trials, there are different subsets of neuron which fire sequentially in time, bridging the gap between the action and the outcome. Now, we believe that mice make the decision to choose right or left somewhere here, way before they make the action, because the start of the choice selective activity signals that the decision has already been made. So, in fact, the cortical inputs bridge the time between the decision and the outcome in the form of these recorded choice selective sequences. We next built a neural circuit model of this task. The cortical inputs to the model are the choice selective sequences that you just saw on the last slide and are shown here in a cartoon form for a right choice trial. The Fs represent the activity, the recorded activity of each recorded neuron. Now, since the left choice neuron's activity is just 0 on right choice trials, we will just focus on right choice neurons for visualization purposes. Now, we believe that these cortical inputs act as the temporal basis functions using which value is calculated over time. Now, since NAC receives these cortical inputs, we believe it carries the temporal value components given by the weighted temporal basis functions. So these weights are what determine the amount of value carried by each NAC neuron. These NAC neurons then converge onto the ventral pallidum such that the ventral pallidum carries the complete value signal. Note that since ventral pallidum receives inhibitory projections from NAC, it carries the negatively signed value signal. Okay, now let's discuss how prediction error signals are generated. So prediction error is given by what is known as the temporal difference error in reinforcement learning theory. It's defined as the error in your previous expected sum of future rewards, which is VT minus 1 here, and the updated expected sum of future rewards given by the sum of the currently received reward RT plus the expected future rewards at the current time, VT. Therefore, to generate an RP signal in the network, the dopamine needs to receive all of these three signals. Okay, so the dopamine neuron receives an external reward input. It also receives the positive value signal as required directly from ventral pallidum, which carries the value signal. Since ventral pallidum carries a negatively signed value signal, as you can see here, and sends inhibitory projections to dopamine neurons, the dopamine neurons receive a net positive value signal. Finally, the dopamine also receives a delayed negative value signal indirectly from ventral pallidum through the VTA GABA interneuron. The negative sign is due to this signal going through this GABA interneuron and is delayed as it has to go through this extra synapse as shown here. Note that this represents this circuit just represents one possibility of several circuit implementations we considered, but all share the same core elements of having this fast and a delayed pathway to the dopamine RP carrying neurons. And here is our complete network diagram generating the value and RP signals, including the left choice neurons. Okay, now these generated RP signals are then carried by dopamine neurons to striatum, where the value learning happens by updating the corticostriatal weights. So these weight updates are directly proportional to the prediction error signal coming in through dopamine and the cortical input signal. So this is how the correct values are learned. Note that, as mentioned before, the decision is made at the beginning of the trial by comparing the values of both choices at that time. Okay. Let us now see how this model run with actual recorded cortical inputs. So I emphasize we put the actual recorded cortical inputs in our simulations. Let's see how it behaves. So what we find is that, like mice, the model is also able to choose left more on left blocks and right more on right blocks. Now, one way to see how the mice or model can perform this task is to see how getting rewarded or unrewarded on the previous trial affects choice on the current trial. This is what we show here on the right side. So y-axis for both these plots represent the probability of returning to a rewarded choice or an unrewarded choice on the next trial. Now, as expected, the model as well as the mice prefer to stick more to the rewarded choice than the unrewarded ones. So then we wanted to generate experimentally testable predictions of our model. So, and to do that, what we did was we artificially activated all the cortical input neurons to nucleus accumbens on a randomly selected 10% of the trials. So basically replace this choice selective input activity with this artificial non-selective activity. Now we replicate this manipulation experimentally by optogenetically activating the cortical inputs to striatum on a subset of the trial. This artificial and choice non-selective input activity will lead to non-selective weight updates, which will cause the model to stay less on rewarded choice, or in other words, switch more on rewarded choice and stay more on unrewarded choice. It's like the activation makes the model partly forget what the outcome was on the previous trial. We see the same results for the experiments. Now, here there is an issue here. So the model is able to switch its choice preference in about 3 trials after the actual block switch. But this requires fast dopamine-mediated corticostriatal synaptic plasticity on the order of seconds or tens of seconds. However, there isn't extensive evidence for such fast dopamine-mediated corticostriatal plasticity. Therefore, we currently are performing experiments to determine if such fast dopamine-mediated plasticity does in fact occur in corticostriatal synapses. But in addition, we would like to propose an alternative model that does not require fast synaptic plasticity. So this alternative model is based on the idea of meta-reinforcement learning. The key ideas of this model are that the weights are slowly trained initially, but they are frozen during the task performance or testing. And the fast block switch learning is achieved by changing the recurrent dynamics, but not the plasticity. So I repeat, we do not change the weights, we only change the recurrent dynamics for the fast block switch learning for this alternative model. Okay, now let's see how our actor-critic network for the meta-RL model works. So in this model, the value and RP signals are generated in pretty much the same way as the previous model, except that the values now are generated using a critic, which is a recurrent neural network. Now, we believe that this critic network could reside somewhere in nucleus accumbens, ventral pallidum, or prefrontal cortex, or some combination of these areas. And note that the critic network, in addition to the choice selective input, they also receive a reward input. Now, this dopamine signal then acts as the teaching signal from the critic to this actor network, which is also a recurrent neural network, plausibly in dorsal striatum or cortex, that generates the action. The actor also receives a temporal sequence input, plausibly from the hippocampus, that tells the actor what time it is during a trial. It also receives the action output from the previous time step. Now, as explained before, we believe that the decision of what choice what choice to make on the current trial occurs at the beginning of the trial. So in our model, this choice is determined by the output of the actor, which then initiates the corresponding choice selective sequence for that trial. And this is our complete meta-RL circuit diagram. Now that we know how the model works, let's see how it performs on the task. So the meta-RL model performs well. Again, it chooses left more on left block and then switches in about 3 to 4 trials to choosing right more on right blocks. It also qualitatively replicates the optogenetic stimulation results from the experiments. So again, switching more if previous trial was rewarded, and switching less if previous trial was unrewarded, same as the data. In summary, we showed that the recorded cortical inputs encode choice selective sequences that solve the temporal creator assignment problem by bridging the time gap between actions and outcome. We then proposed 2 models of learning based on the recorded choice selective input, one in which prediction error updates value by updating synaptic weights, and in the other, the prediction error updates the recurrent dynamics to drive learning. We also tested the role of these choice selective sequences by optogenetically stimulating it. So stimulation disrupted learning by either non-selectively changing weights for one model or by disrupting the recurrent dynamics for the other model. There was no time to show this, but both models also replicate previous recordings in VTA dopamine and GABA interneuron. I would like to end by thanking Mark Goldman and Ilana Witten for their great advising, and Nathan and Julia, they were great with the experiments. Thank you.
Great. Thank you so much, Avinash, for that talk. I would encourage other people to add questions if they have them. We have a question from Allison Hamilos. The question is, if the sequence bridges the gap between event and reward, it seems that you should be able to disrupt the choice by inhibiting the units firing in the middle of the sequence. Have you or anyone else tried that manipulation?
Sorry, could you repeat that question again? I was getting, I could not.
Sure. If the sequence bridges the gap between the event and the reward, it seems that you should be able to disrupt the choice by inhibiting the units firing in the middle of the sequence. Have you or has anyone else tried that?
Oh, by inhibiting it, we haven't tried inhibiting the inputs, but when we stimulated the inputs, we stimulated it all through the trial. But yeah, we haven't tried inhibiting it. That is an interesting thing that we should try and do that.
Great, thanks. Eric Garr asks, how confident are you that optostimulation disrupts action-specific eligibility traces as you suggest, or could you be disrupting something else entirely?
We know that the PL neurons that project to nucleus accumbens, they do not really project to anywhere else. So when we are activating those PL neurons, I mean, we are confident that we are only affecting those inputs, the recorded choice selective inputs, rather than affecting something else in the brain.
Okay, so that answer might address this question, but just in case, from Juliana Chase, she asks, or she says, many glutamate neurons from PL that project to the NAC also have collaterals that go to the dorsomedial striatum. Did you ensure that your optogenetic stimulation was only affecting cells in the NAC?
Yeah, it's what my previous answer basically. We saw. I mean, we know that it doesn't really project to as many other places other than NAC, or that's what I believe. So I don't think it should affect other things.
Okay, great. And one final question from Robert Schultz. Can the model also recapitulate the initial emergence of the reward prediction error from reward time to Q time?
Yes, it does. It does do that. We didn't have time to show that, but yeah, it does do that.
Great. So if there are no other questions, let's thank Avinash again for a wonderful talk and all of our speakers for this session. And I believe there's a poster session next. So thank you again and enjoy the rest of the meeting.
Thank you.